We have recently received funding through ARC Linkage Grant, which is a partnership between Marcroft Grains Pathology, Melbourne University and Syngenta. And we also have some funding from Bayer and Adama to look at fungicide resistance for each of the commercial fungicides that are available on the market. And what we're doing is we take stubble from different populations across Australia. So we've got 100 samples that have been sent in by agronomists and farmers, and we use them to inoculate our plants. So in the bottom of the tub here, we have a tray of seedlings. These have each been treated with the different fungicides. So they've either got a seed dressing or they've been applied with foliar fungicide. We then have the stubble that's hung above the plants and allowed to shower the spores down onto the plants for just over 24 hours. So this would have a lid on it so that whole population is fully contained. And so we do this for each of the 100 stubbles.
So one of the advantages of using the stubble is that the sexual spores are released from these fruiting bodies. So each of these black dots is a single fruiting body, and each one of them releases thousands of spores. So what we do is we wet the stubble up, the spores are released, and instead of screening maybe 100 isolates in a lab, we're actually screening potentially millions and millions of spores with each screen. So it's extremely high throughput and extremely diverse set of isolates that are getting tested by using this method.
So this is an example of a tray that was inoculated 2 weeks ago. Essentially what we have here in the middle here is our untreated control. So this has got no fungicide, and we've got lots of lesions developing on our seedlings as we would expect. So in fact, pretty much all seedlings here have been infected. We've also got Maxim in here. So Maxim is registered as a blackleg suppressor, but doesn't actually give any controlled blackleg in the field. So you can see here each of our seedlings is also completely colonized, and then each of the other punnets represent various chemistries that are available. Previously we'd done a screen similar to this, but only looked at Jockey and found resistance in around 15% of the populations that we looked at. So here's an example of the Jockey. So last time we screened this, we had resistance, and this particular population is also showing resistance. So you can see here there's complete cotyledons that have been colonized by the pathogen and causing lots of disease, which suggests that the resistance, the fungicide in this case, has been overcome. The nice thing is that we're not seeing any resistance to any of the new chemistries. So in here we've got Elevo, Moravis, Aviator, Saltro, Veritas, Prezara and Flutriafol. And so the new chemistries that have been released, we're not seeing any lesions, which is great news, suggesting that we don't have any resistance to those new chemistries before they're being released. So that's fantastic. So the new chemistries that we have are SDHIs and some strobilurins, whereas our old chemistries all belong to the azole class of fungicides, and that's where we're seeing some of the resistance, but not in any of these new chemistries. And then lastly, this is our Moravis, and we're not seeing anything at all coming up on these plants. So this suggests that there's no resistance to these new chemistries that are being released at the moment, which is fantastic news for our growers.